Welcome to another screencast where I'll be discussing more DNA history, the experimental evidence supporting the discovery of DNA as the hereditary material. Today we'll be talking about the experiments with experimental evidence that supports DNA as the hereditary material from Avery, McCarty, and MacLeod. All right, in a previous screencast, we had discussed Griffith, and if you haven't watched that one, you need to watch that one first because Avery, McCarty, and MacLeod pick up sort of where Griffith left off and use his model system, and then they tweak it a little bit. Okay. So in the 1940s, sorry to back up, in the 1920s, Griffith was doing his experiment with the strep pneumoniae and the mice, and he had the living R and the heat-killed S mixtures and the transforming agent. Griffith dies not knowing what that agent was. So in the 1940s, Avery McCarty and MacLeod discover that Griffith's transforming agent actually is DNA and not protein. So at this point, they've isolated the nucleic acids. They understand that what's in the nucleus, the primary components are DNA and protein, and they want to know which one is actually the hereditary material, which one is being passed on. Is it DNA or is it the protein? Which one is giving the phenotype to these cells, to these organisms? So what Avery McCarty and MacLeod do is they take Griffith's basic experiment there, and they have living R, and they have heat-killed S, and they mix it together, and they end up getting results much similar to Griffith's. But what they did, how they tweak it, is they, in one case over here, they put a protease. Now, a protease is an enzyme that will break down proteins. So they put a protease in one. In the other experimental group, they take, again, living R, heat-killed S, which should give you dead mice and give you results similar to Griffith. And in this group, they put a DNase. Right? And so this DNase is an enzyme, in general, that will break down DNA. So the hypothesis, if they're using this protease, so if the hereditary material is a protein and we use a protease, then what should we get? Well, the logic was if the hereditary material is a protein and we use a protease, then will this transforming agent here transform these living R cells into living S cells? The answer should be no. Right? So if the hereditary is a protein and we use a protease, then living R not transformed into living S, and the mouse should live. So this is their hypothesis. They do the experiment, they use the protease, and what do they get? They get dead mice. So they're like, okay, it's probably not protein. But then to be sure that they're just not, that the DNA is definitely it, they have to do this other experiment over here, and the DNase hypothesis reads similarly to that. So if the hereditary material is DNA, and we use a DNase to break down the DNA as a hereditary material, then living R not transformed into living S, and mouse should live. And sure enough, this is indeed the results of their experiment. So again, using the model system and tweaking it a little bit from Griffith there, and they're able then to confirm their original hypotheses, right? That DNA is the hereditary material. Right? Not only that, when they subsequently pulled samples out of these dead mice, do you think that they saw, what did they see? Actually, I can ask you that question. We can talk about that sort of in class. What would they expect to see when they pulled samples, tissue samples or fluid samples from these dead mice, and when they pulled fluid samples from these mice over here, what would they expect to see? So this is, if you understand Griffith's experiment, you should be able to answer these questions, and we can talk about that in class there. Okay? So Avery McCarty MacLeod, zeroing in on DNA being DNA being the hereditary material and not protein. Okay? And this classic experiment lends evidence and support to those conclusions. So again, you should be able to describe here experimental evidence, the experimental setup for Avery McCarty MacLeod here, and again, it was based on Griffith. Thanks for listening, and try to make those predictions, bring that information to class, and we'll see you then. Thanks for listening.